The cell cycle includes interphase, mitosis, and cytokinesis. Mitosis has prophase, metaphase, anaphase, and telophase, and the result is identical daughter cells. So when we look at this kind of a diagram, the overall cell cycle contains interphase, which takes by far the longest amount of time. It has G1, S, and G2 phases. We have mitosis, which again is prophase, metaphase, anaphase, and telophase. And then when the cell splits, we have cytokinesis. When we look at this, we can also have cells go into a G0 phase where they're no longer dividing and they're just kind of existing. Now, with the right prompts, G0 phase can be interrupted and it can go back into the overall cell cycle. Another thought too is that some cells are actually, if mitosis is division of the nucleus, we can actually go through mitosis in multinucleated cells without actually splitting into separate cells. So overall, we have the interphase, we have mitosis, and we have cytokinesis. These little stop signs are checkpoints, which we'll talk more about in the next topic. So when we have our cell cycle, when I teach it, a lot of times I talk about spaghetti and sausages, because in this process of cell division, if you had a plate of spaghetti, it would be very difficult to separate each individual strand. Whereas if you had sausage links, you could separate them quite easily into two separate plates. So initially, we have our chromatin, which is very loose, and you're able to access the actual DNA and access the genes. Again, a lot of times when we think of DNA, we think about the double helix, but sometimes we also think about chromosomes. Now, during interphase, where the cell is growing, the DNA is copied, and the cell grows some more, we have replication taking place. This is the time when we end up with more chromatin, and it's more like spaghetti and looser, and the DNA is still accessible. When we enter into prophase, I sometimes call it, that's when we get our sausages, because the DNA coils up into tightly wound chromosomes. And again, it's at this time when you can see that sister chromatids that had been copied during the S phase. So during prophase, the chromosomes coil up, the nuclear membrane breaks down, centrosomes migrate, and spindle fibers begin to be sent out. At metaphase, meta's in the middle. So at metaphase, the chromosomes are lined up in the middle of the cell, making sure that the sister chromatids are split equally to each of the daughter cells. At anaphase, we start pulling these things apart. So the chromosomes, the sister chromatids, begin separating away from each other. In telophase, we have two. We have two nuclei within one cell. So the nuclear membranes start reforming around the DNA. Still one cell, but during cytokinesis, it is officially split, and we go back to that growth phase. We can't continue to divide and divide and divide without growing. Otherwise, our cells would get so small that they wouldn't have all of the machinery needed to exist. So the whole thing is a cell cycle, and mitosis is only the division of the nucleus, which includes prophase, metaphase, anaphase, and telophase. So again, interphase, the longest phase, broken up into G1, S, and G2. Prophase, we get our sausages. Meta's in the middle, ana's apart, tele is 2, and cytokinesis is split. So as we go into this a little bit more, again, this G1 phase, the cell is growing, the chromatin is loose, transcription translation is happening, protein synthesis is occurring, and the S phase is when the DNA replicates. So that's where we get our sister chromatids, and even though it's loose, it's still going to have an extra copy of all of the chromosomes. At the G2 phase, the cell is going to continue to grow, continue to exist, and the protein synthesis that's happening during the G2 phase is to create the proteins necessary for division. So this early prophase, sometimes we break up the prophase into early prophase, and prometaphase. During the early prophase, we have to ultimately also duplicate the centrosomes. So the centrosomes are duplicated. We started to condense the chromatin into chromosomes, and we're starting to lose and break apart this nuclear envelope. As we get into prophase, we don't have a nucleolus anymore, which was generating ribosomes. So the nucleolus is gone. You can see the duplicated sister chromatids. You can see them together. The centrioles or the centrosomes are starting to migrate towards the poles, and they're starting to send out spindle fibers. In prometaphase, you have the centromere, where we have the sister chromatids attached, kind of wound into each other, but we have the kinetochores starting to be attached to spindle fibers here. So the kinetochore are little protein clusters that are located at the centromeres of the chromosomes. So in metaphase, everything is lined up in the middle, and this is a really important checkpoint because it's at this point where we kind of have spindle tug of war and make sure that spindle fibers are attached properly to the kinetochores before they start dividing. Otherwise, we can end up with non-disjunction. So in anaphase, we actually do get some separation here. The sister chromatids are being pulled, and they're kind of being walked as well through kinesin and dynein motor proteins along the spindle fibers, and the spindle fibers are also shortening as they go. So in telophase, we start reforming the nuclear envelope, so it's still one cell, but we have two nuclei within that cell. We start getting the nucleolus to come back. Again, that's our ribosome factories. And the chromosomes start unwinding back towards chromatin, kind of going back towards spaghetti. So in cytokinesis, they split, and we have this contractile ring and this indentation that's occurring at the cleavage furrow. But this contractile ring is made out of cytoskeleton actin proteins. And again, that's kind of squeezing it apart, and eventually we split. If they're plants, we have a cell plate, but cytokinesis is when the cell actually splits. So again, during the cell cycle, we have interphase, mitosis, and cytokinesis. Mitosis is broken up into prophase, metaphase, anaphase, and telophase, and the result is identical daughter cells.